When you start looking at toggle triad, so as we discussed, this is a more frustrated system. So you would get these three dominant states. But interestingly, what you notice is that if you take any pairwise correlation between AB, AC, or BC, the correlation coefficient decreases to almost half of what you saw in a toggle switch, because if any of these two is strongly antagonizing each other, then you would not see the third state coming in. So that's the compromise that it reaches, that the antagonism weakens down such as to allow for these three states to eventually give rise to. So then the question comes that how does this dynamic change when you embed this in a large network? And this work was led by Pradyumna, who was then a master's thesis student at IISER Bhopal and has now joined us for PhD. So what we have done here is we take different external networks of different sizes, 5 node, 10 node, 15 node, and 20 node, and also different densities. So you could have 10, so number of edges is equals to twice the number of nodes, number of edges is equals to 4 times the number of nodes, or 6 times the number of nodes. So you have these 12 combinations here. And for each of these combinations, we have taken 100 networks each. So here, for instance, you have 100 networks, all of which have 5 node and 10 edges. There can be many, many combinations like this. So we bootstrapped and took any 100 combinations and then embedded in this a toggle switch or a toggle triad. The condition for embedding was simple, that whatever you're embedding should have each node of that embedded motif should have at least one incoming edge. It can be activation, it can be inhibition. That we did not control for that. Okay? So now you have 1200 such networks. And we again simulated using the same strategy I was talking about yesterday, because we do not have exact parameters. So you sample over a large parameter regime and then see how the behavior changes. Now, what are the metrics? So one metric we used is this correlation coefficient. So for the unembedded network, the correlation coefficient is minus 0.8. So we asked, how does this correlation coefficient change? Because now let's say if you're looking at a network here, you have a 5 node 10 edge plus 2 node 2 edge. So you have a 7 node 12 edge network. So it's a 7-dimensional network. You simulate it over large cohorts of parameters and then take all the solutions and ask what is the correlation coefficient between A and B. Now, you could, what we've done here is, now let's keep the density fixed and look at across different sizes. And what you see here on the y-axis is a correlation coefficient, and these are distributions of those 100 cases each. So when the density is fixed and you vary the size, just varying the size doesn't really give you a significantly different result as in terms of correlation coefficients. But if you do the opposite, you keep the size the fixed and you increase the density, you see a trend. The higher the density, the weaker the correlation coefficient between A and B. So the unembedded is somewhere here, minus 0.8. So clearly, as soon as you embed it, you begin to see values which are higher than the unembedded case, which means that the mutual antagonism or exclusivity between them is weakening. And you can even see that in some cases it reaches 0, right? So depending on which network we are looking at, A and B are just completely uncorrelated. You have equal number of solutions in first, second, third, and fourth quadrant, and that's what makes it the correlation coefficient very close to 0. So what this tells us is that the density matters more than the size. That's the first message. And then we measured it using other metrics, bimodality coefficients of A. So bimodality coefficient is defined again on a scale of 0 to 1. Anything above 0.6 using this metric that we have adopted from previous literature can be called as bimodal. And the higher the value, the more clearly distinct the solutions are. So again, what you notice is that when you vary the density, keeping the size fixed, you see that bimodality coefficient continues to decrease. For the unembedded one, the value was somewhere around 0.75. And so all the embedded ones most of the embedded ones have lower values than that. And again, the higher the density, the less distinct the two solutions of A are. So next we wanted to ask, was it the global density or was it the local density which was impacting the behavior? So this is one example. Again, we have plotted correlation coefficient of between A and B. X-axis, here this is in this diagram, this is the in degree of B, how many nodes are coming onto B and how many nodes are coming onto A. So this is the control case when A and B have both one input each. This is the unembedded case because A is being inhibited by B and B is being inhibited by A. So clearly the correlation coefficient is minus 0.8, and you could clearly see if you go along this diagonal, at a stage when there are 5 additional nodes coming onto A and to B, the correlation coefficient drops sharply, and you are at minus 0.3. And of course, you could vary one of them as well and see these differences. Now, this is where we wanted to look at more carefully about how the symmetry versus asymmetry of incoming nodes on incoming edges on A or B made a difference. So here is an example. This is the unembedded case. In degree A is 1, in degree B is 1. So this is just A and B. And correlation coefficient is close to minus 0.8. And F1, which basically means number of solutions in second and fourth quadrant, is around 0.9. Now, when you do this scenario where you add 1 more in degree to A and 5 more incoming edges to B, so you notice that the behavior of A is not changed as much. It is reasonably still bimodal, but the behavior of B has completely changed. It's no longer bimodal. But when you compare it to another case where the total in degree is still the same, 2 plus 6, 8 here, 4 plus 4 is also 8, you see a very different behavior that both of them have changed their behavior. The bimodality is completely lost. So it depends on the local density, it depends on also how many edges are coming onto each of the nodes rather than just talking about the overall motif here. Okay, and this is just a summary of those, that it depends on in degree of A, in degree of B, and therefore it begins to depend on the total in degree as well. Then we embedded toggle triad into this instead of a toggle switch, asking whether the same trends are observed here as well, and the answer is yes. When you vary the, you keep the density fixed, vary the size, doesn't make a difference. In this particular case, interestingly, as far as you're looking at correlation coefficient between just A and B, even the density doesn't make a difference. So this is when we realize that just looking at correlation coefficient between A and B, when you start looking at a toggle triad, is not necessarily the best metric. So we then, because here in this case, you could have 3 correlation coefficients, AB, BC, and CA. So what we asked was, maybe information is not contained in correlation coefficient between AB, but it is contained in BC or AC. So let's take the maximum value of the correlation coefficient, right? So that is what max CC, max of these 3 values. And again, you begin to see a trend. And again, these values, interestingly, they even they reach all the way up to plus 0.5. So when you embed a toggle triad, you begin to notice that, any 2 nodes, we don't know which one it is here because this is the maximum value of those 3. But it begins to show positive trends as well. So toggle triad could begin to have, the antagonism can convert into the activation kind of behaviors here. And again, F1, which is the number of states with 1 positive and the other 2 negative or low, it also decreases as a function of density, but not as a function of the size. So the next question we asked was, okay, what happens? So let's say that when you embed a toggle triad, there are limits in which you no longer see that a toggle triad behaves as a toggle triad, or F1, which is the frequency of states where 1 is high and the other 2 are low, or single positive states is decreasing. But what is increasing? Is it the double positive states which are increasing? Is it the triple positive states or triple negative states? Those are the only 3 possibilities. So what we noticed there was that indeed the double positive states begin to increase, and there's not really a whole change in the triple positive states. They continue to stay below 10% of the steady state distribution. And interestingly, if you plot a ratio of single positive to double positive in the limits of higher density and higher sizes as well, you begin to see that limit is reaching 1, which basically means that as you embed a toggle triad, what begins to happen is that the difference that we notice between single positive and double positive, that single positive was almost 1.5 times the frequency of double positive, that difference disappears, and now you have all 6 states, the 3 TH1, TH2, TH17, and also the hybrid states, TH12, 217, and 117, they all begin to appear at a similar frequency. That's the only change that is happening. So embedding them seems to be enriching for those hybrid states. That's the qualitative change we are having. The next question we asked was, what if there is self-activation or self-inhibition? Because many of these master regulators have been shown to have usually self-activation. So when you do that, so this is the blue is the simple toggle switch or simple toggle triad, orange is the one with self-activation added, and green is the one with self-inhibition added on all the nodes of a toggle switch or a toggle triad. So again, what you notice is that the blue and orange are very close to each other, but green is the one where the behavior is most disrupted. So if you have self-inhibition, then that leads to a faster decay of the properties. But if you have self-activation, it doesn't really help you a whole lot. Okay. So that again explains at least the observations that why some of these master regulators either have self-activation or don't have any self-regulation, but usually they do not have self-inhibition. And the possible reason behind that is because when you embed them in larger networks, you would see a faster decay. And evolution may have chosen for networks such that the resilience to embedding them in larger networks is still maintained. But when you add self-inhibition, the decay is faster. This is where we actually started looking at networks published and well curated in the literature. And we found that at least in the E. coli network, the average in degree per node is less than 3. And if you go back to the this plot, so if you look at 3 and 3 here, you're still above negative 0.5. So you're still seeing reasonably negative solutions here. But of course, even at 4 you do that. But beyond 4 nodes per 4 edges per node is when you begin to see that toggle switch no longer remains a toggle switch if you set minus 0.5 as the cutoff. Okay, so this possibly explains why. So what this tells us is that as long as you have around 4 nodes per 4 edges per node coming in, you're still okay. And that is what the literature suggests, that in that example, in the real system, actually average in degree per node is less than 3. So that could possibly explain why these toggle switches continue to behave as toggle switches even when you embed them in larger network. Their dynamics or their mutual exclusivity patterns may be compromised, but that compromise is not so high that you would say that this is no longer a toggle switch. Okay, now let's look at some large networks. So this is one of the examples. This is, suddenly we have switched from 3 nodes to 33 nodes, okay, an order of magnitude higher. And this has 357 edges. So doing ODE-based simulations for this one was computationally quite expensive. So we switched to a very simple Boolean models. And Arijit will talk in much more detail about Boolean models here. So I'll just mention what is relevant here. So in Boolean models, in this case, every node can have just 2 values. +1 or -1, on or off. Okay? There are no parameters in this case. And because we do not know the exact rules that each node follows, so let's say one node has 5 incoming edges. Is it an AND gate? Is it an OR gate? Is it a more complex combination of any of them? We do not have that information. So we adopted to a very simple majority rule. We said that for every node, let's count the number of incoming edges. If the number of activatory incoming edges are more, you turn the node on. If the number of inhibitory edges are more, you turn the node off. And if the number of activations and the number of inhibitions is equal, you do not update the node. If it was on in the previous step, it stays on. If it was off in the previous step, it stays off. Okay, very simple majority rule. Now, for a simple toggle switch, we notice that, so our simple toggle switch is, you have 2 nodes, 2 edges, and you predominantly get 2 states. For a 3-node, 6-edge toggle triad that we looked at, you also get 6 states. For a 4-node, I think the toggle
tetrahedron was 12 edges, you also got predominantly 6 states. So what are your guesses for the number of states in this one? This is toggle switch, this is toggle triad, this is toggle tetrahedron. Anyone would take a guess how many states would we see here? How many of you think this would be greater than 100 states? Greater than 1,000 states. Let's start as high as we can. Greater than 1,000 states, okay? Between 100 and 1,000. Okay? Between 10 to 100. Less than 10. So the answer is 4 states. We saw only 4 states in this network, which could, which corresponded to 99 point something percentage. So in this, each column is one of these nodes. White is on, black is off. And you see the top 4 states actually correspond to the most 4 ones. The other, we plotted the top 10 ones, and you could clearly see a distinct drop in the frequency. So we were also very surprised that how is this possible that in such a higher dimension network you have such a few number of limited states, right? So we did this analysis where we said, let's take this network and let's swap the edges between them. Okay, so you're not changing the size of the network, you're not changing the density of the network. All you're doing is changing the connections between them, and then ask how many states we get. So we did this 1,000 times, 1,000 unique non-biological hypothetical networks, and this is the distribution on a log scale. So you can clearly see there are networks where you get 10,000 states or 1,000,000 states, but this is where our wild type network lies, which has only 4 states. So there's something very, very special about the topology of this particular network, which is leading to such a dramatic reduction in the steady state space, which was not seen in any of the other networks. The next question we asked was, now let's take these 357 edges and delete one edge at a time, or change the sign of an edge from positive to negative or negative to positive at a time, and ask, does the behavior change? And the answer to that is, so this is the change of sign of an edge, and what we are measuring is Jensen-Shannon divergence, which basically tells you how far the 2 steady state distributions are. 0 means they're completely overlapping, 1 means they're completely non-overlapping. So the x-axis is when you change the sign of an edge, the y-axis is when you delete an edge. So this single dot that you see here, if you zoom in further, these are 345 cases. Only in 12 cases, which are somewhere here, you see that there is some change in the phenotypic distribution. In the remaining 345 cases, deletion of a single edge or change of sign of a single edge, one at a time, leads to no change in phenotypic distribution, suggesting that the network is reasonably robust, at least to single edge perturbations, right? So we started thinking about what are so special properties about this network that it leads to such high robustness and it leads to such a dramatic reduction in the state space, right? Now, this is where you could again draw parallels with developmental scenarios, right? We have at the end of the day around 200 to 250 cell types in our body, right, which all originate from the same cell. But how many possible interactions are there? How many different possible interactions are there, right? So if you think of, say, 20,000 genes and say 30,000 proteins in each cell, and you only allow for, say, only these networks. So if you have a 30,000 cross 30,000 interaction matrix or adjacency matrix, you could have such a huge number of networks emerging from there. But that comes down only to 200 cell types. So there are some mechanisms which are canalizing this dramatically, such as to reduce the possibility space in a major manner and allow for only a limited number of states, right? Now, we'll come back to this network in a minute. I'll introduce another, and by the way, this network is in the context of small cell lung cancer, which is a highly, highly aggressive cancer, exclusively associated with smoking. The 5-year survival percentage, 5-year survival rate of this cancer is less than 10%. So it's a highly aggressive cancer, and we still do not have any targeted therapy for this. We use the usual chemotherapy, radiation therapy, and combination of that here.
In a diseased cell, you see a network which is not part of your healthy body emerge, which has these properties. And also, have you looked at networks in healthy cells in a similar manner?
Right. So that's a great question, right? So what I was asking is how in the developmental cases, it's not surprising that you have these scenarios, but what changes happen in a diseased case? So we have some very preliminary evidence suggesting that in diseased states, you actually begin to see many more states as compared to what you saw in healthy states. So this paradigm does get broken to some extent in a disease state. And yes, but there is a catch in the way this network was identified. So this network was identified using statistical methods. So we are not even sure if each node is well curated or not. So that's a catch, and that's why. But later we looked at the examples at present, where the networks have been built bottom up. So each node is well curated, and there we also see similar behaviors. So just to introduce the context of cell state transition, other examples we'll be looking at in a minute. So metastasis, as many of you may know, continues to be the major cause of all cancer-related deaths. And what has been shown or what has been realized by the field over multiple years is that in the context of metastasis, there are no unique mutational signatures that you could say drive metastasis. So mutational signatures could drive tumor initiation, initial steps of it, but in the context of metastasis, what cells would eventually go to the other site and proliferate and grow a colony there are no unique mutational signatures known. And metastasis happens to be a highly inefficient process, thankfully. Only out of 10,000 cells, about two cells are able to eventually grow a tumor at the second site. So what has been realized now is that it's the phenotypic plasticity, or the ability of cells to switch their behavior or switch their phenotype in a reversible manner, which is important for metastasis, because there are two advantages that this dynamics offers, which mutations do not offer. First of all, this is fast, right? So a cell could change its phenotype rather quickly in response to its environment. If suddenly the oxygen levels go down, you could switch your metabolic behavior to a different behavior. But for mutations, you would have to wait for multiple cell divisions to occur so as that you could get this. So again, at a fast time scale, phenotypic plasticity or phenotypic adaptation; at a relatively slower time scale, genetic adaptation plays a role. And also, these changes are reversible in nature. So if you're thinking of a multi-stable system where cells could switch back and forth between the different states depending on the perturbation that you give, unlike mutations, where mutation, once a change has been done, it will be irreversibly passed down to every single cell in that lineage, and you cannot get rid of that mutation and change. So phenotypic plasticity has been identified more recently as one of these hallmarks of cancer, and what could happen as a consequence of this. Again, an example we discussed yesterday. So let's say you have a cell population which you could classify as bimodal based on the levels of all given protein of interest. You segregate these populations and you think that these are homogeneous population which will stay like that for a longer duration. But when you culture them independently, invariably you see that the other population also comes up, because again, the other state exists in the system, and at some some value, some rate which is dependent on many parameters, you could begin to see the emergence of the other, right? So if you're thinking of this in the context of cancer, let's say you have drug sensitive and drug resistant cells, you have, let's say, segregated them, and again, you think that the drug sensitive cells will stay drug sensitive. That's not the case. Some of them will give rise to drug resistant cells. Eventually, of course, I'm not going into the quantitative details. You may get similar to parental behavior, you may get different than parental behavior. All that depends on multiple parameters. So one of the most well-studied phenomena in metastasis is something called as epithelial to mesenchymal transition. Now, if you think of the most common solid tumors, say breast cancer, prostate cancer, lung cancer, liver cancer, etc., they are all epithelial in origin, which means that cells are stuck tightly to their neighbors. They do not have the intrinsic ability to migrate or invade. But then they gain these properties, which are usually the properties of mesenchymal cells in our body, and they begin to migrate and they breach the basement membrane, they reach the blood vessels, and they're able to enter the circulation for metastasis. Now, once they reach the metastatic site, they no longer need to invade, they no longer need to migrate, they can actually settle down, they need to settle down and grow a whole tumor there, which is the exact opposite transition called MET. So this EMT and MET is what continues to drive the cycles of metastasis. And again, going back to Rahul's point, EMT and MET is not something that cancer has invented. These are normal processes that happen multiple times during development. And again, Ramray can speak much more about this than I do. So there are, for instance, in the formation of heart, there are 3 or 4 cycles of EMT and MET that cells go through to be able to form these different layers of the structure of a heart. Okay. Now, EMT or MET was largely thought of as a binary process almost a decade ago. And some of the work we did initially making mathematical models predicted that EMT was not a binary process. You could have this third state where cells are neither completely epithelial nor completely mesenchymal, which is what we called hybrid states. And there was one other paper around the same time which predicted the same things, looking at a slightly different network, and suggested that this could be a stable phenotype that cells can maintain in addition to completely epithelial and mesenchymal. Now, the implications of this are strong because these cells had a different phenotypic behavior, which is collective migration, because you have not completely lost adhesion, but you have gained the property of migration and invasion. And in the past few years, it has been now shown that collective migration is the predominant mode of metastasis when cancer cells leave as clusters instead of leaving as single cells. So over the past 10 years or so, what people have identified is that yes, these hybrid cells exist, and you could have more than one hybrid states, and these hybrid states are actually essential for tumorigenesis. If you get rid of these hybrid states, tumor formation goes down. And interestingly, these hybrid states have been suggested to be most plastic or most easy to switch from as compared to completely epithelial or completely mesenchymal. So the current paradigm is that yes, you have these multiple states in between, and those states happen to be most aggressive, most drug resistant, most metastatic as compared to extreme epithelial or extreme mesenchymal, because they are most adaptive. Okay. So the question we then started thinking was that why are these cells most adaptive? Right? Can you explain this based on the network structure? Okay. So sure, you have to improvise, adapt, and overcome. But where is this high level of adaptive behavior coming from? So Kishore, my PhD student who's now a postdoc at Northeastern, he started thinking about this, and we gathered multiple networks which people had built in the field of EMT over the years. So we started with a 3-node network 10 years ago, but now these are much larger networks. And again, going back to Rahul's point, these networks are constructed bottom up. So what different colleagues in the field have done is they have added one node at a time by looking at relevant literature. So these are not identified in a statistical manner. So the chances of a false positive are quite low in this case. Okay? So nonetheless, you have these networks, and we look at the results of one of the networks in more detail, but we saw very similar behavior in others as well. So let's focus on this one, where because we now extensively know which nodes are epithelial, which nodes are mesenchymal. What I mean by that is that there are these nodes which, when overexpressed, you could drive EMT, and these nodes, when overexpressed, you could drive MET. And this has been well established in the literature. Now, when you simulate this network using the same strategy, you again see something very similar to what was noticed in the small cell lung cancer case. That there are a bunch of states where all mesenchymal nodes are on and all epithelial nodes are off. A bunch of states where all mesenchymal is off, all epithelial are on. And this, I don't have the numbers here, but this corresponds to, again, approximately 95% of the steady state space. And in the remaining 5%, you see these so-called hybrid states, where some epithelial are on, some mesenchymal are on, and they can be again in different combinations. Okay. So we then define these two metrics. One is called frustation, which is again directly taken from spin systems, which basically means that how much a state disobeys a network structure. Okay, so basically, if you look at epithelial and mesenchymal, because most regulations, if you go back to the network, most regulations across epithelial or mesenchymal are inhibitory in nature, shown by red, and most regulations within mesenchymal happen to be activatory in nature. But of course, when you will have scenarios when some of these states will not obey this behavior, they all happen to be hybrid phenotypes. So the hybrid phenotypes are indeed more frustrated. So which means that explains why it is easier for these cells to get out of it. But still, it does not answer the question completely. The question still remains, why are hybrid states more frustrated? Okay, what we have done so far is just took a metric of frustration and said that, look, hybrid states are more frustrated. That is why they are most plastic. But why so? Okay. So now let's go back to this network and let's represent this network in the form of a matrix, where each row is the source, each column is the target. Now in this 22 cross 22 matrix, only 82 elements are filled, which means they are either +1 or -1. +1 is activation, shown by red. -1 is inhibition, shown by blue. And the remaining 484 minus 82, which is 402 edges, they don't exist. Right? So it's a very sparse network in that sense. But as we very well know that in networks, if let's say A activates C, which impacts B, then A is also impacting B indirectly. Right? But that information is not captured here, because this is capturing only adjacent interactions, right? So if if we were to draw a matrix for the interactions that I just mentioned, A to C would be +1, but A to B would be 0. But we know that C is also passing down information to B, which is missing here. Right? So how do you capture that information? You capture that information by multiplying this matrix to itself. So that now you capture all, you take one step and then you take the other step and so on. So now if you multiply this matrix to itself many, many times, and of course normalize it by the path length. So the longer the path length, the less the weightage given to that path. When you do that, you see an interesting behavior, that every epithelial node effectively, when I say effectively, I'm basically taking all the paths that go from one node to the other node and collapse them onto one path. Every epithelial node effectively activates every epithelial node. Every mesenchymal node effectively activates every mesenchymal node. Every epithelial node effectively inhibits every mesenchymal node, and every mesenchymal node effectively inhibits every epithelial node. So it's a very simple structure. You're back to a toggle switch. It looks like a very complicated network, but you're back to a scenario where basically it's something similar to this at the end of the day. Now, instead of one A, this is what we call, there is a team of A. Or instead of one B, there is a team of B. But that's the only difference, right? Now the question comes that, can this then explain why we saw such a reduced dimensionality space? Okay. And again, we did the same analysis. We asked, if you randomize the network edges, if you generate non-biological hypothetical networks, what happens to team strength? So you could define team strength as basically an average of these four numbers, of the mean of these four numbers. Magnitude of these four numbers on a scale of 0 to 1. So this is where the wild type network lies. It's around 0.7 on a scale of 0 to 1. And this is the histogram of 1,000 such networks. And you can clearly see only a few networks. I think wild type was around 96th percentile. So 95% of the networks do not have that formation of teams. So this is again something specific to the topology of this network. And what are the implications? The implications are very clear. What we did here is, with the question we were asking is, if you start from an epithelial node, if you start from an epithelial state, mesenchymal state, or hybrid state, how many edges on average would you need to flip? How many nodes on average would you need to flip from off to on or on to off such that you can get out of that state? So what is the minimum perturbation needed to push that initial state out? If you start from mesenchymal or an epithelial case, that answer is 50%. If you start from hybrid, that answer is 5%. So there's an order of magnitude difference in the relative stability of these states, which could directly be explained by the presence of teams, because if you look carefully, teams are only present for epithelial and mesenchymal. There's no teams for hybrid states. So the hybrid states, those poor guys get pulled in one direction or the other as soon as they come out. But for an epithelial state to become a mesenchymal state, or a mesenchymal state to become an epithelial state, there is strong resistance being offered by the presence of these teams, which is there in the structure of the network. So then we went back to the small cell lung cancer case, and we simulated it and looked at pairwise correlations. So again, you see this phenomenon of teams showing up. You could also do the influence matrix calculation. So this calculation did not require any simulations. All you have to do is just multiply the matrix. You're not even simulating the network, and you could still identify that there is a structure present in the topology of the network. But when you simulate it, and you can clearly see this phenomena, that there is this team of 22 nodes, this team of 11 nodes. And when you look at real data, this is data taken from Cancer Cell and Encyclopedia, and you take the same 33 variables there, and these are around 50 samples here. You see a very similar behavior to what was predicted by the network. You can change the cancer, you can change the nodes. This is melanoma, a different cancer. Again, you see this team's behavior predicted by the model, and you see a very similar behavior shown in real data. Okay? So that suggests that the dynamics of these networks could indeed capture what is going on in real data. The next question we started thinking about is, can teams be, a dimension reduction metric? So, usually when we talk about TSNE or UMAP or these kinds of plots, so of course it helps us reduce the dimensionality and see how the data looks like, but it doesn't really give you a whole lot of information of which variables are contributing the most there. Then of course you can project your favorite molecule and see whether they are different or not, but it doesn't tell you where it is coming from. So what we did here was, we took this network and we simulated it using ODEs this time, because when you're trying to do principal component analysis, then using discrete outputs is not the best way forward. So we did this, and to our surprise, we found that principal component 1 could explain 95% of the data. And again, you could look at non-biological networks here by randomizing the edges between them, and this is the distribution of principal component variance. The mean is somewhere around 0.4, and the wild type network is 0.9. And now if you look at those 100 networks, non-biological networks, and you plot this distribution, plot this scatter plot, where the x-axis is the team strength that you could calculate before doing any simulations, and the y-axis is principal component 1. So you could see this neat sigmoidal, Michaelis-Menten kind of kinetics behavior here, that the stronger the team strength, the higher the principal component variance. And this is where the wild type network lies, far away from all the other non-biological networks. Now, interestingly, when you look at the coefficients of principal component 1, because what does principal component do? It basically is a linear combination of variables, which explains the maximum variance, right? So you could actually look at the coefficients of that and identify which variables are in the same direction